Okay, so we are going to introduce now Paul Zamora. So first of all, can you see the slide, the lung cancer biology overview?
I can.
Okay. All right. So Paul is our chief scientist for this project, but we do have a large team, and we're always looking for other members. So this is community-driven science. Uh, I'll hand it over to Paul. Paul, if you could give a little bit of introduction on yourself and your background.
Sure. My name is Paul Zamora. I am a retired scientist. During my work career, I was associated with a number of small companies that were focused on radiopharmaceutical development uh, or uh, projects involving medical devices, including those that were combination medical devices for use in orthopedics. So I oversaw the research and development of uh, several projects and products leading up to early clinical trials. Matthew?
Thank you. And then I'm Matthew Zamora. My background is I was undergraduate for neuroscience, but since that time I went into more of a technical realm. So I've been a web applications developer. But I love this project because it kind of brings me back to my biology roots. So with that said, why don't we go ahead and get into it? And first up, we'll send it over to Paul.
Okay, thank you. This introduction is a very broad-based introduction, and it is mainly targeted to a very broad, non-knowledgeable audience. So if there's people that have specialties in lung cancer, this is probably not targeted to you. I want to start first by giving you an overview of what the normal lung is and what the normal lung does. The normal lung is part of the respiratory system, and its primary function, arguably, is oxygen and carbon dioxide exchange. It has two primary components: a conducting system that is a series of tubes that descend from the mouth and trachea down to an exchange area that is involved as a region in actual oxygen and carbon dioxide exchange. Histologically, if you take a section through one of the primary sections, which is the bronchioles toward the more distal end, the more exchange surface oriented end, you can see a couple of things that are of interest. First, you see a big hole in the middle. That's the airway. And then one of the things that is labeled is the epithelium. The epithelium is the lining of cells around that hole. Also, underneath that, you see another area that is marked as smooth muscle. Smooth muscle is found pretty much all the way down, starting from the smaller bronchi all the way down to the alveoli. And for anybody who's ever had asthma, you would know that the inhaler drug that you use, like albuterol, is targeting these smooth muscle cells. It is also targeting the epithelial cells, allowing them to secrete less mucus. And both of those two things in concert allow the people who have asthma to breathe better. On the next slide, Matthew. Okay, the deep lung is associated with a series of, or an alveolar region. The alveolar region is largely composed of a series of very small sacs, very thin, where the carbon dioxide and oxygen exchange actually takes place. The airway tubes connect from the outside of the body all the way down to the deep lung, and the alveolar structures are surrounded by a very rich blood supply area that allows a relatively facile exchange of oxygen and carbon dioxide. Next.
Well, one thing before we move on, I think we want to make very clear the structure, the morphology of it is for surface area. You're really optimizing for the gas exchange by maximizing the surface area. But yeah, I'll go ahead on.
So this is a cross-section, a histological cross-section through the deep lung. And the first thing that is obviously noticeable is there's a lot of holes. On the left-hand middle, you see a relatively large tube that is a terminal bronchiole. And that leads into several areas where there are, for example, the lower right-hand corner, marked as alveoli. These are sections through small sacs where the oxygen exchange actually takes place. If you count the number of branches from the main stem bronchi just below the trachea, all the way down to the respiratory bronchioles, all the way down at the end, there's about 17 to 19 branches. So the branching in the lung is extensive. And that leads down to the gas exchange area. And Matthew had pointed out that the surface area is important, and it's absolutely correct. The rate of gas diffusion in this area of the alveolar area is related to the surface area and also the thickness. The thickness here in the region of the alveoli is very thin, and the surface area, because it's composed of lots of individual sacs, is quite large. In any human adult lung, there is about 300 million of these little sacs called alveoli. So those can represent an extremely large surface area if you take into consideration the whole lung. On the next slide. Bit of background on tumors and cancers. Tumors and cancers are abnormal proliferations of cells that occur in the body. Tumors may be either benign, that is to say they're not cancerous, or they can be cancerous, that is to say they can be malignant. Tumors are clonal in nature, that is to say that they arise originally from single cells that begin to differentiate in an abnormal way. Cancers are diseases of abnormal cells, and they divide without control and can invade other tissues. And that is really the defining characteristic of cancers, lack of control and invasive capabilities. They can also spread not only locally, but distally, far away from the main source. And they do that primarily through the blood and lymphatic systems. They metastasize. Just a note, there's more than 100 different distinct types of cancer, and they vary considerably in their behavior as well as their response to treatments. And only malignant tumors are actually properly referred to as cancers, although many people use the terms interchangeably, even in scientific settings. Next. What is cancer? Well, cancer is a multi-step genetic evolutionary process. It involves multiple serial mutations, that's the genetic component, and cycles of selection for specific cancer cells, that's the evolutionary process. These two combined, the serial mutations and the cycles of selection, lead to progressively increasing capacity for cell proliferation, survival, invasion, and metastases. Cancers, especially the solid cancers, are not genetically uniform. They have substantial intertumoral heterogeneity. They vary from place to place. If you start at the outside of a tumor and you go to the center of it, the core, the gene expression can vary quite dramatically from those two positions. For example, the DNA content and the chromosome number are frequently very different and not normal. non-normal chromosome number is very common in many tumors. And very importantly, cancers evolve solutions to selection pressures. That includes therapies, that includes the body's own immune system trying to fight them off. They evolve, they solve problems, unfortunately. The way that they do this is by engaging a multiplicity of oncogenes as well as a variety of different mutated tumor suppressor genes. And we'll be talking more about both oncogenes and tumor suppressor genes in a little bit. Next, Matthew. Just a bit of background, some demographics. This is specifically has to do with lung cancer. Worldwide in 2022, the most recent statistics available, there's about 20 million cancer cases that were newly diagnosed, again, in the world. And in the same year, it was estimated there was approaching 10 million people who died from the disease. By 2050, the number of newly diagnosed cases are expected to increase substantially to 35 million, based solely on projected population growth. These numbers, of course, can be driven down if one variable is changed, and that is smoking behavior. In the United States, in 2014, there was about 125,000 deaths. New cases in 2024, something approaching 250,000 new cases. In 2021, again, the latest statistics that I could find, there was an estimated over 600,000 people actively living with lung cancer in one form or another, at one stage or another. Lung cancer is the second most common cancer both in men and women. The average age at diagnosis is 70, but it can vary quite widely. Black men tend to have a higher rate of lung cancer than white men. Black women have a lower rate than white women. Hispanics are sort of in between, tending more toward the Caucasians. Death rate, fortunately, in the United States has been decreasing over time, so that there basically has been, the rate has been cut in half since 1992. And a large part of that is due to a decrease in smoking behavior. Next. There are types of lung cancer and carcinomas. Just a definition before we get too far along. A carcinoma is a cancer that forms from epithelial cells. An adenocarcinoma is a cancer that arises from epithelial cells of glands, and in case of the lung, these are mucus-producing cells. Most lung cancers arise from lung epithelial cells. These are the ones, again, that line the surface of the airway, those tubes that I'd showed you earlier. Histologically, there's two major types. Small cell lung carcinoma, which makes up a minority percentage of the lung cancers. It has a neuroendocrine origin, and it normally is involved in making hormones that control the flow of air and blood in and out of the lung. Small cell lung carcinomas, making up to 85% of all lung cancers, and includes adenocarcinomas, make up the balance of the other main set of tumors, lung cancers. And again, these also arise from the epithelial lining of the cells, of the airways, excuse me. There are some subtypes associated with both small cell and non-small cell lung carcinomas. They're illustrated here, and Matthew, I think, will touch on those in the next slide. Matthew?
Thank you. And before we move on, I want to introduce a convention we're going to start using for the rest of this talk. Because there's two major types, there's the small cell cancers and the non-small cell cancers. We're going to use green for small cell and blue for non-small cell, these kind of shades here. It's just so we kind of keep track of the two categories of cancer that we're talking about. It's a little bit of a convention. So we'll continue then. So I wanted to talk a little bit about the two types of lung cancers. So if we're looking at here is a histology, which is it's a staining. There's a biopsy, and you take a small sample, you put it on a slide, you stain it. The black dot, or so the purplish dark dots are going to be the cell nucleus, and the pink is going to be the interstitial fluid or the cell wall. So what you can see with the small lung cells are you have these very round nuclei. Over on the non-small lung side, they're very squished, and that's a very common phenomenon with the non-small cell lung carcinomas. They do have that kind of distorted shape to the nucleus. Going to the small lung cell again, small lung cells are primarily involved with neuroendocrine cells, which are found in the bronchi, the air tubes leading from the trachea to the lungs, and for the non-small cell lung carcinomas, there's the adenocarcinomas. This again originates in the mucus-producing cells found in the outer parts of the lung. It is the most common form of lung cancer in both smokers and non-smokers. Squamous cell carcinoma, another one for non-small cell lung cancer. It begins in squamous cells, which are flat cells lining the sides of the airways. It often is associated with a history of smoking and typically forms in the central part of the lungs. And then large cell carcinoma, also known as undifferentiated carcinoma. This type can occur in any part of the lung and tends to grow and spread quickly, making it more challenging to treat. So with cancers, there's 4 stages conventionally, and they are kind of rule of thumb associated with the size of the tumor. So we're looking at stage 1, which is less than 4 centimeters, or approximately the size of a walnut, a little bit less. Stage 2, you're getting to that 4 to 7 centimeter, so it's going to be between a walnut and a peach-sized. Stage 3 is going to be a lime size, and perhaps, and then stage 4 is going to be about the peach size, 7 centimeters or more. The stage 2, generally you're going to have the tumor has migrated to nearby lymph nodes, and stage 3, it's already migrated to the lymph nodes and maybe parts outside the lung. And then stage 4 is it's spread to other organs, or it's larger than that 7 centimeters. So where do the lung cancers arise? Again, we're talking about those 2 big categories, the small cell and the non-small cell. The non-small cell, we're seeing them up here in the trachea, the primary bronchi, and the small cells are going to be kind of in that intermediate area, and then again the non-small cells all the way at the bottom with the bronchioles and alveoli. You'll notice the category for smoking is very predominant, especially in those upper sections of the lung. All right, we have a question from William. What's your question?
So, what is a small cell? Why is it called a small cell?
The question is, what is a small cell and why is it called a small cell? It has to do, I think, with the parts. So when you talk about an organ is going to be a mixture or a composite of many different types of cells. We are talking about just different parts of the body. So we have, for example, where in the system those small cells are involved. But it's just a naming convention for the different parts of the tissue. Paul, is there any other nuance?
It was based originally on pathologists looking at slides like you have imaged here, and under a microscope it appeared that many of these had a differential size, associated both with the nucleus and the overall cell itself. And since early on, when people were looking at this, size was an easy way of differentiating between structures. Things that were small, since there was a number of them, came to be associated with a naming convention. You would note here that Matthew was talking about large cell carcinomas on the other side. Again, these arose just by people looking under a microscope at these and seeing that they were larger than surrounding cells. So it's just a naming convention that's kind of stuck around for a while. And is actually very widely used and accepted in medical terms. They do have different functions, however, and different things that they arise from. Matthew?
Thank you. So that's all I have for this one. We'll proceed on to this one. Thank you, Paul.
Okay. So if you ask the question, when is lung cancer detected? And what happens in terms of detection and survival? Non-small cell lung cancer is usually detected by stage 3. Stage 3, it's already metastasized. Small cell lung cancers typically are diagnosed at stage 3 or 4. Again, they've already metastasized. The reason that metastasis is so important, and I've been stressing it, is that the 5-year survival rate, if it's localized, that is to say it's not really metastasized, as compared to the 5-year survival rate if it has metastasized and is found in a distant organ, is shown in the table here. Where you look at the distant 5-year survival rates, they're, regardless of the cancer, it's below 10%. This is a dismal rate. So early detection would be a clear benefit in lung cancer. And having metastases is clearly not a favorable outcome. Next. Where do the lung cancers actually metastasize to? Matthew mentioned that they initially go to lymph nodes that are close by. They can also end up in distant lymph nodes. The brain, bones, liver, adrenal glands, and other parts of the lung, or in the other side of the non-infected lung. Nonetheless, regardless of where they end up, the 5-year survival rates are truly dismal. So anybody that starts off with a lung cancer that shows signs of metastases, their survival rate at the end of 5 years is extremely poor, regardless of where the metastasis actually ends up. Next. Part of this has to do with the growth rates of the tumors. So a note again that malignancy is a frequent occurrence in both small cell and large cell lung cancers, and it occurs early. Non-small cell lung carcinomas grow faster than small cell lung carcinomas, and they frequently, again, metastasize before diagnosis. The tumor volume doubling time is on the range of 4 to 5 months. That can, again, vary quite widely, but that's just a sort of a working average. Small cell lung carcinomas are quite aggressive, and they, again, metastasize before. And their tumor volume doubling time can be as short as 1 month, although, again, that average varies widely. But they're aggressive tumors. Next. Screening would be nice. So early detection would be nice. Unfortunately, there's only one test that is recommended for lung cancer, and that is low-dose computed tomography. It does fortunately have a very high sensitivity and specificity, but it is only now currently being recommended for adults who are at high risk of developing the disease because of their smoking history and age. I did, however, want to give you at least a view of what a radiologist might see in one of these scans. And the arrow points to a metastasis, or to a primary tumor in one of the lungs. And this is again done with computerized tomography, or CT. Because CT might be a bit new and unfamiliar to you, I thought I would just take a second and digress, Matthew, on the next slide, and give you an idea of what is computerized tomography. Computerized tomography uses a combination of X-rays and computer technology to produce images of the inside of the body. It takes an X-ray beam and moves it in a circle around the body, allowing different views of the same organ. The X-ray information is sent to detectors, which process the data and display it in a two-dimensional format in the form of slices starting from the head and going to the feet. The successive slices can be digitally stacked and then reconstructed in longitudinal images or made into 3D images. This is computerized tomography has made a basically revolution in detection of all kinds of diseases, not least of which is lung cancer. Next. There are, however, several other diagnostic imaging techniques that are used for lung cancer. Chest X-ray is one, is the least effective. Its sensitivity is moderate, but it is still widely used because it's cheap and easy. Computerized tomography is the most sensitive technique. It does have false positive values that are low, at least on the initial screens. Magnetic resonance imaging, MRI, is also used, and so is positron emission tomography. Positron emission tomography is based on the use of a radioactive tracer that is used into the body, and that tracer is then taken up into the cancer and subsequently imaged. There's also a combination of PET and CT. The image on the right-hand side illustrates a PET CT with the yellow being highlighted as the primary tumor and then the metastases further down. Next. There are several treatment options that are available for lung cancer, and they fall into two main categories: traditional medicine and precision medicine. Traditional medicine, by that what is meant is surgery, standard chemotherapy, or radiation treatment. Precision therapy is a targeted therapy, and we'll be talking more about that, and also immunotherapy, which is something that we will largely not be talking about, but it is coming to the forefront in medicine as well. Next. One of the things that is important to note is that while a person with cancer may have treatment options, those options will change as the disease progresses. So if a patient has limited disease, that can be addressed directly with surgery. On the other hand, if they have extensive disease, including metastases, the types of treatment options that they have shrink considerably, so that chemotherapy and targeted radiotherapy are some of the only options that are actually available. This again points to a real need to try to address early detection. On the next slide. This is a list of some common chemotherapy drugs that are used to treat cancers. They're not quite the same. There is some overlap between the non-small cell lung carcinomas and the small cell lung carcinomas. In the non-small cell lung carcinomas, typically oncologists use a platinum-based therapy, and that would include cisplatin and carboplatin, used in one or the other in combination with one of the other drugs that's listed below. So paclitaxel, docetaxel, any one of those in the list below. They also use others, but these are the most common ones that are actually used. So again, a combination therapy. Small cell lung carcinomas do not typically use cisplatin, but they do use combination therapies as well. And these are some of the drugs that are used there. Next. While there's a whole range of drugs that are actually used, they tend to fall in several classes, generalized by a mechanism of action. Typically, that involves either DNA binding, DNA synthesis disruption, microtubule stability, and DNA repair. Microtubule stability is associated with the inability. When you target that, what you're trying to do is stop the cells from dividing. They need microtubules to separate from one cell from the other during division, and disrupting that frustrates the cell and they end up dying. Next.
All right, thank you. I have a comment here. It seems to me that we, as a group researching, possible compounds for cancers, we're interested in really getting down to these groups, these mechanism of action groups, or other ways of grouping these compounds. I think one of the concepts here is, especially in the context of combinational therapies, if we go after one route, like for example, the cisplatin is this DNA binding mechanism of action group. If we are looking for possible co-therapies, I would think we would want to target a different group with that co-therapy. Paul, is that your understanding?
Well, yes. What is used in standard medicine, for example, is to combine cisplatin with a microtubule disrupting agent like paclitaxel. They have two different mechanisms of action, and that's really what you want to do. You want to be able to spread your toxic effect across a number of pathways to try to get as effective a death coverage as possible. So attacking the cancer cell from two different directions, as it were, provides a potentially better outcome. And it actually can help in decreasing the dose and making it less toxic overall for the patient.
And thank you. And it seems to me that our group, like these categories, we're using the term very roughly mechanism of action. I think one of the things I want to highlight for our group is that there are different ways of bucketing these compounds, and we want to think of what those other ways of bucketing compounds are. Which part of the biochemical pathway are they targeting? That's an important one. Structurally similar compounds, that's another way of looking at it. But there is this idea of approaching within a group what compounds are kind of clustering around what they are defined as, and then also looking laterally what are other types of groups, because we know about this co-therapy context. Let me go ahead and move on from that. Targeted therapies are used mainly for advanced non-small cell lung cancer, and they target specific proteins that cancer cells need to survive and spread. So this is based on biomarker testing. We're looking for detectable changes in the tumor DNA. Those are mutations, alterations, deletions, rearrangements. The drug target specific protein changes. drugs are used to target specific protein changes, and the protein that you're going after is the target. There's a preferential interaction with tumor cells compared to normal cells. So we're looking at drugs that have some activity level in the tumor cells more so than normal cells. So if you're targeting a protein, you want something about that protein to be more meaningfully expressed in the tumor cells versus non-tumor cells. Maybe there's too much promotion going on, or maybe they're multiplying so quickly and they have a strong reliance on critical cell division pathways. That kind of concept. But there needs to be something different about the tumors that you're going after. This is applicable to a subset of patients that have that appropriate target. So when you have these drugs and you're developing them, they're intended for something very specific. So we'll go through this example here. Osimertinib is indicated as a treatment for patients with locally advanced, that is unresectable, not able to be surgically removed, or metastatic non-small cell lung cancer, whose tumors also harbor EGFR, epithelial growth factor receptor mutations, specifically exon 19 deletions or exon 21 substitutions. Okay, well that's a big wordy thing to say, but what we're saying is this drug was designed to target something very, very specific, right? And that's going after even specific mutations. Just because a protein is mutated doesn't mean it's not effective. It really depends what is the mutation and where is it in the structure. There's definitely an upcoming need for more and more personalized medicines. It is fair to say that when you have people with even small cell lung cancers, that there is not only heterogeneity within the tumor itself, but also within populations of people. They're not all going to respond to the same classes of things. With that said, I did want to call out a few of the genes. I do this not because I expect you to remember it, but just because repetition and exposure as we go into these topics I find to be very helpful. So with targeted gene therapy, we have growth-promoting cell surface receptors, VEGF, EGFR, ALK, ROS1, MET, RET, and the growth-promoting signal transduction, KRAS, and BRAF. So with that, I'll hand it back over to Paul.
So in this particular set of therapies, biomarker testing is mandatory, although it is, quote, recommended for people with advanced non-lung cell cancers. The purpose is actually to guide targeted therapy options. You need to point out also that there are no FDA-approved treatment options based on small cell lung carcinoma biomarkers. So when we're talking about lung, you're really talking about only one group. It happens to be the major group, but nonetheless, only one group. This biomarker testing is typically done by taking a fine needle biopsy or a sample from going down into the lung airways. It's called a bronchoscopy. Or in some cases, a liquid biopsy is taken, and by that it's meant blood. And there you're looking for circulating tumor cells or circulating tumor DNA. This is primarily used to monitor metastases. So in biomarker testing for lung carcinoma, there's a number of approved and targeted biomarkers and subsequent targeted therapies, and there's a list very similar to what Matthew had presented before, so I'm not going to go through it. On the next slide, you can ask the question, does it work? And the simple answer, the short answer is yes, it does work fairly well. So if you look at progression-free survival, that is to say, the tumor is not growing, and overall survival in patients who have received either targeted therapy as compared to those with only chemotherapy, the graph that's presented here demonstrates that the 2-year survival rates for overall survival is quite higher in patients that receive targeted therapy. There's basically a doubling in overall survival rates. So it does work. It does improve survival. That's the good news. The bad news, the people are still dying. There still is a very high level of mortality, even with targeted therapies. To make things even a little bit more complicated, targeted therapies are only going to be applicable in perhaps up to 30% of all patients. In lung, it's probably even lower than that. So if you only 30% of the patients are eligible for targeted therapy, and not all of them survive the therapy procedure itself, there clearly is room for some kind of therapeutic improvement. On the next slide.
I want to take a minute for a question. Eric asks, are blood-based methods for early cancer detection effective?
Depends on the type. Not in lung cancer. There are a couple of screening tests and other tests that have been developed and are frequently used for colon cancer, ovarian cancer, in particular. Those are the ones that I know of and can remember. But there's none available for lung cancer.
Okay, thank you. We have another question?
Yeah, why is the eligibility for targeted therapy so low, like 30%?
The question was, why was the eligibility for targeted therapy so low, like 30%? Let me start with, I think I'll let you answer that, Paul. Go ahead.
Because only a certain percentage of the patients have the mutation necessary to allow the therapy. You have to use biomarker testing to show that they actually have a mutation that can be affected by the drug that you want to use. If they don't have the mutation, you can't use the drug. So you start cutting down on the total number of patients because not all of them have the appropriate mutation.
But I think the counterpoint is, if you do have the mutation, you might have something specific you can go after that, as shown in this chart, may be a little bit more effective than otherwise conventional chemotherapy. Okay, so I want to talk a little bit about how lung cancer can use the system to develop resistance. And in fact, it's going to recruit a number of mechanisms to enable that survival. So there's a few things that can become hijacked or that can be dysregulated through, again, that genetic evolution of the cancer that will promote the most aggressive malignant forms of the cancer. We have DNA repair systems. Things that would normally fix mutations can be turned off, or things that are regulating the quality of intact DNA sequences can be misregulated. Drug efflux, pro-survival signaling, cell cycle arrest, what's killing it, what's stopping it, epigenetic regulation, things like methylation, epithelial mesenchymal transition. Paul, what's that one?
That is a revision back to a stem cell characteristic.
Thank you.
So
tumor microenvironment. What is the kind of the signaling chemicals around it, or if there's inflammation signals, those kinds of things. Drug metabolism, how quickly things are able to be extruded or removed. and then the cancer metabolism itself, the ability to recruit more blood cells and just take over the energy production process. So these are very animate and very active, but also dynamic systems, right? Things that change over time, and certain patients could have certain of these more prominently playing a role than others. And this also goes back to lead into the next slide. This goes back to what the specific genetic mutations are. You might have some things that are making some of these pathways a little bit more or less relevant for the cancer. So I want to talk a little bit about tumor suppressors and this new concept of synthetic lethality. So synthetic lethality is something that we're introducing here that we want to bring to the table to the, kind of the cancers conversation. And it's this idea that you can have a normal cell, and then if you have a corresponding a normal cell, and then at some point, for example, that normal cell could have a mutation that causes the cancer. So the A in this diagram is the thing presumably causing the cancer, driving the cancer. So the cancer driving mutation. Now, if you have that mutation, it's possible to target that mutation, right? That is a conventional chemotherapy. But if you have a coincidental genetic gene, if you have a specific gene that is coincident to it, you might actually be able to target that. And that might actually strongly increase your ability to target that specific cancer. So this is the concept of synthetic lethality, is that you're able to use these kind of other lateral genes in support of the ability to target the actual gene that is ultimately driving the cancer. So the survival of cancer cells within inactive tumor suppressor gene A, which it's a loss of function to the gene A. The thing that was suppressing it, you're losing that. And its ability to do what it does depends on the expression of gene B. It has this kind of lateral dependency. So if you inhibit gene B, it leads to the synthetic lethality. It kills the cell through this other process. So a little bit about gene B could be DNA repair genes, cell cycle control genes, transcription regulation genes, and others. I'm going to take a pause there for Paul. Paul, is there anything else you want to add to this?
No, actually, I think that's fine.
Okay. A little bit about the background of this synthetic lethality concept. It is actually known for a while. It was described in Drosophila in 1911, and it's defined as the co-occurrence of two genetic events that result in organismal or cellular death. It looks like our call is about to end. I'm going to post another link just in case. Give me a second. Sorry, I didn't realize that was going to happen. All right, so let's go ahead and get back into it. So we had been talking about synthetic lethality. It is a relatively new concept, as we're seeing here. In 1997, it was suggested as a cancer therapy. So they used synthetic lethal interaction partners for cancer-associated molecular changes. Okay. 2005, it was applied specifically to tumor suppressor and DNA repair genes, BRCA and PARP inhibitors. The clinical trials for that was 2010. And the FDA did a really surprisingly fast expedition of that approval to 2014, and this was for women with advanced ovarian cancers. So again, holistically in the cancers, synthetic lethality is relatively new, but it is definitely something that we're thinking of leaning into in our upcoming manuscript as far as something we want to use. So with that, I will pass it over to Paul, but before we do, we have a question from Eric, and I'll pass this up to Paul. Since the cancer is evolving quickly, how many measurement time points do you typically need for synthetic lethality techniques?
Oh my God. Unknown is I think the real answer. You want to start the therapy as soon as you have a definitive proof for it. In the case of BRCA is something that is known to be associated with a high rate of lethality in women. It's associated with breast cancer and ovarian cancer. And there had been not a whole lot of good treatments for patients that have this disease. So as soon as it can be diagnosed and an appropriate proof established for the absence of BRCA genes, and loss of function in the protein, you can get started. Will they change? Yes. Will the loss of function come back? No, it will not. So that once you lose function, the component is not coming back. I hope that helps.
Thank you, Paul. All right, we'll go ahead and continue to the next slide. Paul, this one's for you.
Okay. So we've gone back and we've looked at synthetic lethality in lung cancer specifically. And most of the efforts that I've been able to track down have been associated with the screening component. And they've largely focused not on a tumor suppressor, but on an oncogene. And that oncogene is, the name of it is called KRAS. Tumor suppressor genes appear to be under-evaluated. On the next slide. There's probably a very good reason that many of the investigators have focused on KRAS as an oncogene, as it might relate to synthetic lethality, because it is present in so many non-small cell lung cancers. This pie chart illustrates that there about 29% have a very high rate of mutated KRAS. Next, Matthew. Back to definitions. What is an oncogene and what's a tumor suppressor? An oncogene is a gene that is found in cancers that has, I'll start again. An oncogene is a gene that has the potential to cause cancer. In normal cells, it is usually referred to as a proto-oncogene, something that comes before the oncogene. Oncogenes promote cell proliferation, or they inhibit apoptosis. Apoptosis is a cellular process that is associated with a programmed cell death, which is actually normally a very important function. It helps to regulate cell proliferation and keep the balance of cell growth and cell death in check. Oncogenes are often mutated and expressed at very high levels in tumors. There's a number of oncogenes that are upregulated and found in high levels in lung cancer. This is a list of some of them that you see in the next line there, and it's sort of the usual suspects. We've talked about some of these before. Tumor suppressors, on the other hand, these tumor suppressor genes act to negatively reflect cell division or promote apoptosis. They're downregulators. They're brakes. Inactivated tumor suppressors lead to an uncontrolled cell division and an increased cancer risk. In the previous study that we did, we had focused on one tumor suppressor, NF1, and that's part of that has in some part led to our continued interest as we move forward. So NF1 is actually found as a tumor suppressor in lung cancer as well. On the next slide, Matthew.
All right, so I wanted to take a minute to kind of reiterate what we covered with the tumor suppressors So again, the analogy here is this car pedal, the gas, and the car brake. So the tumor suppressors are acting as brakes. In a normal situation, when you have active tumor suppressor genes functioning, they're going to downregulate that cell division or whatever is moderating the cell growth. And then the proto-oncogenes in their normal position, they are accelerators. They're used. You have wound repair and you have normal functioning cell division, and so you have them, but they're not going to be all the way down. If we look at the right two columns, when we have this mutated state, effectively this is the analogy. The tumor suppressors are like you're missing the brake, or there's nothing turning off the ability, the signaling. If you have it like a cut and you have these growth factors there, the growth factors are signaling, but you don't have any counter growth factors. You don't have anything counteracting that kind of positive signal. You can see how that could get pretty bad. The oncogenes at this point are the gas, and the gas is all the way down, right? We are signaling, cell growth, that kind of thing. So that's kind of your analogy for the tumor suppressor versus oncogenes. The tumor suppressors are this brake which can become defective, or the oncogenes are this gas pedal that might be all the way to the floor in some cases. So with that, I'll push it back to Paul.
Okay. So if you just take a look at the significantly mutated genes from an adenocarcinoma, lung adenocarcinomas. Again, adenocarcinomas are related to non-small cell lung carcinomas. You just look at the total frequency, and what I've done here is use red arrows to indicate tumor suppressors. And you see that the number one mutated gene is a tumor suppressor. It's called TP53. And there are a number of other tumor suppressors that are indicated here as well. Now, KRAS, as we were talking about before, is sort of the second in the series. But nonetheless, P53 is the most, which is a tumor suppressor, is the most mutated gene in lung adenocarcinomas. And on the next slide, you see that is somewhat mirrored when you go to small cell lung carcinomas. Again, the number one gene that's mutated in this cancer type is TP53, with a frequency of almost 90%. Another tumor suppressor, RB1, is mutated in these types of cancers at a rate of about 64%. These are very high numbers. So not only do oncogenes play a role and are frequently altered, but tumor suppressors play a role, and they are frequently altered. And this may provide, again, a new way of looking at ways to regulate tumor growth. Matthew?
Thank you. So I do want to take a minute here to call out TP53 and RB1. If you see how they're so highly ranked on this chart for small cell lung cancer, it's going to be no surprise to you why in our paper coming up we're focusing on those two. So we'll get into that coming up on these slides, but this is a rationale for how we're picking some of our focuses, and it's just the frequency of these mutations. So looking ahead, I want to go through briefly the pathway for what might be some of these oncogene growth pathways. So you have, for example, up here, you have a variety of receptors. This could be the epithelial growth factor receptor. And then if we're talking about where is KRAS coming in, it's connected to that receptor as part of a protein complex. All of these orange dots are the are different proteins, and they're all bound to this receptor. And in fact, when the receptor is signaled, this complex activates a cascade of downstream activity as well. So from the RAS, we have this RAF, MEK, MAPK pathway, all of this going into this gene transcription cell cycle progression. We don't talk about it here, but we had mentioned P10, PI3K, and AKT. They're also involved in this cascade. But looking again at these key genes, the EGFR is up here. The RAS plays a role up here, and it's a very early signaler. So having it mutated so often means that it has a very strong effect in these tumors. And then this BRAF that we were talking about briefly is also one of those early in the cascade. So let's also talk a little bit about the pathogenic pathways in lung cancer. We're looking at the growth suppression and the tumor suppressors. So here, for example, we have the role of two tumor suppressors. NF1, which we kind of highlighted briefly, is some of our past work in other cancers. As we mentioned, it does play a role in lung cancer. It's not maybe the most common, but it can play a role. P10 also indicated here. And so this symbol is, of course, the inhibitory symbol. So it's playing that breaking role. And likewise, if NF1 is mutated, then your break is gone, and you can see how that cascade would also result in the cancer. Over here on the right, we have the mitogenetic stimulation, which is the role of cell division. And actually, I'm going to let Paul, if you could talk to this one a little bit.
Okay. NF1 and P10 act very close to the beginning of the cell signaling process. There are other places in the cell that tumor suppressors can play a role, and that is closer to the nucleus. And those are represented by P53 or TP53, as I've called it before, which acts as a transcription factor directly, and RB1, which we've talked a little bit about before, which when it reacts with a transcription factor called E2F. But these are very close to the nucleus and interact directly at the level of the DNA. So it's a slightly different mechanism, but they end up the same thing happens. You get uncontrolled growth and decrease in apoptosis, regardless of which you're using. Something that is close to the nucleus affecting transcription factors, or something that, on the other hand, like NF1, is affecting signal transduction very close to the cell membrane. Matthew?
Thank you. And I think these little black labels, G1 arrest, S entry, these have to do with different phases during the cell division process. So you kind of see a progression from one phase into another phase. That's kind of what the mechanism is implying. Okay, so again, as far as just some name recognition, the tumor suppressor genes with two mutations, tumor suppressor genes that have homozygous mutations, that is one mutation in each of the two alleles, they generally express proteins with loss of function, and protein doesn't work anymore. And these loss of function mutations are cancer drivers. So we're not going to go into this table deeply. I did want to, again, make that connection. TP53, it's involved in apoptosis. The RB1 is a DNA replication, cell division, cell death. And then we touched a little bit on NF1, cell division differentiation, that RAS pathway, and the BRCA, cell division and death. So just again, making some of these associations that we'll want to look at deeper in our upcoming literature reviews. All right, so that's a lot. We have a planned break here, so let's take maybe just a 1 minute break, and we'll get back into it in a second. Okay. I actually see a question from Eric from a while ago. Sorry for missing it. Eric asks, it's not clear the difference with what about an oncogene? Is an oncogene an otherwise normal gene but has undergone some mutation making it dangerous? Or is an oncogene a normal gene causing division? I would say this is kind of going back to when does a proto-oncogene become an oncogene? A proto-oncogene is normally helping with the cell divisions, normally doing its active ability involving in the cell division or cell survival process. It could also be on the apoptosis side, so it could be that it is normally killing the cell in response to cell signals you're supposed to. So it's either division or survival in some way. And that's the proto-oncogene state. The definition of the oncogene would be that same gene but mutated and attributed to what's probably driving the cancer, right? So this fact that something with the mutation has now become misregulated, and the thing it was doing before, which was a good thing, now it's doing the wrong thing. You know, whatever that is, whether apoptosis is no longer killing the cell, or the cell division is just on overdrive. Does that answer your question, Eric? Okay, thank you so much. Let's go ahead and continue then. Into some of the genes that we're focusing on for our research. Paul, I'll hand it over to you. Oh, Paul might be away. Paul, are you with us? You're muted.
This is just to reiterate after the break that there are a number of tumor suppressor proteins in lung cancer. The protein called p53, or also known as TP53, is one of the ones of major interest to us because it is found in high frequency in lung cancer. It functions in response to DNA damage and helps regulate the cell cycle. Another one that we're quite interested in and have decided to focus the paper around is RB1. RB1, and there's some other ones, including P16, that are involved in pathways for the regulation of the cell cycle. We've talked about NF1 and P10 as being associated with signal transduction pathways, and ultimately, through that signal transduction pathway, controlling cell division. The next slide. Just to reiterate again that our focus is going to be on just a couple of these genes, being TP53 and RB1, and that is largely driven by the high incidence and high frequency of mutation in lung cancers. So this is a slide that we've seen before. Next. Okay. One of the things that has been known in the literature for a while is that the gene called RB1, which is a tumor suppressor, has a very close association with another gene, separate gene called aurora kinase. And cells lacking the RB1 tumor suppressor gene, I'll just read it off of the title, are hyperdependent on aurora kinase for survival. This was noted in one of the first papers that described RB1 and its role that might be linked to synthetic lethality. Synthetic lethality was described for an RB1 tumor suppressor gene about a year later. And the title of that reads, Aurora A kinase inhibition is synthetic lethal with the loss of RB1 tumor suppressor gene. So this has been established. There had been established an association with RB1 and synthetic lethality, but not in lung cancer. This is in a different cancer. These were actually associated with a type of throat and type of throat cancer, oral cancers, as I recall. On the next slide.
Thank you. So, wanted to talk a little bit about the function of aurora kinases. So, it's involved in cell division. What you have here is a phase of cell division where you are splitting the DNA. You have that chromatin in the middle, which is the lined up DNA, and there's a splitting. And those green lines are the microtubules, and they're physically being split and ultimately creating two separate nuclei, leading into these two separate cells. So you're going to see that aurora kinase plays a role in this cell division process. It's affecting the chromatin, the centrosomes, which are those splitting mechanisms, and the new cell cortex. And then this is just briefly, we're going to focus on the the yellow circle, but this is where the RB1 fits in. In the diagram, it's just called RB, but this is the RB1, and it has to do with this cell arrest cycle. So we may recall that we had talked earlier about RAS being very early in the cascade, and we see it at the top here. And there's this RAS cascade that leads to a later interaction. Now, this shape doesn't mean anything. It's just saying that there are these multiple routes of signaling. There's these two branches of signaling leading into the cyclin D, and they're both relevant, right? Cyclin D is moderated by more than one thing at once, one of them being this RAS, for example, and this RB then also having this role, again, as described, I think, near the nucleus and near the cell division process with stopping the cell division process. I'm going to pause there. Paul, is that about right? Is there anything I missed? Paul, you're muted.
No, that's fine.
Okay, thank you. All right, so we're going to switch gears now. That's the majority of our biological basis for lung cancer. What we want to talk about now are the bridges between that work, that foundation context, and our current work. So what I want to talk about is basically the algorithm that the Computational Biology Working Group is working towards and has been working on. And this is something that we're looking to publish in the context of lung cancer, specifically because lung cancer is a really well understood cancer. We're looking at that small cell lung cancer, we're looking at that TP53, that RB1. We know they play a role. We can really well define what those roles are. And then we want to be able to essentially say, well, what compounds can be used to get that kind of effect that we want, this ability to go after the RB1 or the aurora kinase and the TP53. So the first thing that we're going to do is we're going to run a panel that, if you have a bunch of compounds that you're testing, you're going to run a screen and see which you're going to see which compounds affect which cell lines. So these are cell lines. Maybe they're biopsied from a tumor. Maybe they're cloned, and then you're able to do high throughput screening. In any case, you kind of know what the responses are going to be. It's assumed that we have this data about what a compound is doing to a specific cell line, and that cell line obviously expressing a specific and presumably known genetic profile. We know what's mutated about it. So once we have that data, say we have all the data where we have all of the compounds and all the different cell lines, we are now going to try and answer questions about what treatments could be. So we're going to focus now on selecting a type of cancer and a type of tumor suppressor. So we're going to insert here, lung is a type of cancer, and tumor suppressor it could be that RB1, for example. So with that selected, now we're looking across this large data set. And by the way, this large data set in our study is DepMap. There is a high-throughput screening study called PRISM, which has very, very large amounts of data, thousands of cell lines, thousands of compounds, and many, many hundreds of types of cancers. So we kind of have the data coverage, at least in the study that we're looking at. So we're gonna subset those two cell lines. One of them we're going to have all the cell lines with 0 mutations. This is the RB1 kind of wild type population, and the other set of cell lines that have 2 damaging mutations. If 2 of them are gone, then we know that's a loss of function for this tumor suppressor. So we have these 2 cell line populations. Within that population, we're going to derive a value S, which is it's a single value that represents how effective a compound could be for that type of cancer, at least as we observed it in a high throughput screen. So it's taking all of the information about what its activity is and kind of giving you a single value. Ultimately, S is a ranking metric. You can rank compounds against each other by looking at this S in indice. So we're taking the S for a given compound, and then we're creating the mean of that. We're saying we saw 10 cell lines that had 0 mutations, and for paclitaxel, it did this. Paclitaxel is a chemotherapy. And then for that same compound, when you had this knockout set of, it had this other average response. Then we're going to look at this pooled mean value, which is just saying we're looking at the difference between what did it do in this wild type, the situation where this tumor suppressor was fine, and what did it do in the population where the tumor suppressor was mutated. And if presumably if you're seeing something going on in the tumor that's not going on in the normal tissue, that's meaningful, right? And that's in fact what we want to go after. Again, looking for that difference between what's going on in tumors versus non-tumor. So when you get that delta S prime, which is just what we're calling this final ranking metric, you are able to then identify which of those compounds in the context of that cancer and relative to that tumor suppressor that you're looking at, which of those compounds is inducing more of a sensitivity to cell death or more of a resistance to cell death. And this allows us to ultimately come up with a comparison mechanism and a ranking system. So just to go over this a few more times, we're going to start with our raw data. In this case, our raw data was that screening data. We have a dose response curve, which is a kind of sampling of how much activity a given concentration is causing in a cell. The activity for chemotherapy is cell death, right? We're giving it a chemotherapy. We're seeing how much cell death in a population of cells did this chemotherapy give. And as you increase the concentration, you would expect to see more and more cell death. This gives us a curve, and this is ultimately giving us data that we need to create our S algorithm, our S prime. Now, the dose response curve data alone isn't enough. We also need to understand the mutations, right? We're really getting at this, we're leading into this algorithm of saying what's the population that has this like RB1 mutation and what's the population that doesn't. So there's a joining of the data sets where we're looking at the response for a given cell line, but it is assumed that we can join in the mutation data for that cell line. Like we know that cell line does or does not have the RB1 knockout. Now we're basically at this point getting our delta S prime. We see that over here. We're finding the relative differences, but we can start to answer questions about groups of compounds. We're going back to this mechanism of action. We have these sets of compounds that all have the same mechanism of action. We would expect that all the compounds within a mechanism of action, if one of them is a tumor suppressor, they should all be, or if they're all effective chemotherapies, everything in that realm, relatively speaking, should be a chemotherapy. I'm sure there's some ups, some downs, and maybe some that do the opposite of what you expect. But on average, for that group of compounds, you would expect it to have a meaningful signal of either it's creating a drug-sensitive or drug-resistant outcome. So once you have this kind of relative difference, and you've already you've implemented into your analysis how you're bucketing these compounds, whether it's by MOA or by target or however you're bucketing it, ultimately what we care about is creating a ranking system. So there's this ability to rank, compare. You're looking at what's the drug sensitivity, what's the drug resistance. For a given cell line, maybe you could optimize for which compounds might be the most effective. This starts to get into that personalized medicine and also the concepts of the targeted therapies. So a little bit more details on the dose response curve that we brought up. What you have here is, as you increase this concentration, this is in a log space, but as you increase the concentration, you expect to see more cell death. So this on the left, on the y-axis, is the response, right? And so the response is it's the cell death, right? And we're assuming something like a luminescence assay where, how many cells light up, say how many there's alive. So if there's less lit up, then less are alive. That's how we're kind of interpreting this. So all of the black points are individual concentrations, and they're sampled for their activity after maybe like a 4-day incubation period. And essentially you're able to put it into this formula and get a theoretical curve. It's a sigmoid curve. It kind of represents the overall effect of what that compound is doing for that cell line. And the technical term here is 4-parameter logistic function model, and we are basically taking that curve and extracting some key attributes of it, the A, D, and C values, this A being the upper asymptote, D lower asymptote, and C, which is this concentration at 50% of the activity. It's the inflection point for this curve. We're using those 3 values to kind of represent what this curve is doing, and we're actually, in the next slide, we're going to see that we're going to collapse those values. So we're taking the overall activity of the chemotherapy. We're using this formula up here, active asymptote minus lower, and we're collapsing it into the single value here. Now, in our original published work, which was just the S by itself, it was delta S, but we didn't have the prime part. It was using just a standard log space. So actually there's a little bit more nuance here that in our paper we have the full formula. But essentially it was a standard log space. This one, which is arcsine, is actually defined as a log-like function. One of the differences here is that we ran into a problem with the original where for example, if the curve that we were looking at here were to go up, we wouldn't get a good value, or if the curve was relatively flat, like we weren't seeing a strong effect, we kind of had these illogical values. It couldn't handle the negatives and the near zero values well. This new function does. So what we're doing in the lung cancer paper is we're introducing this newer S prime algorithm, and we're validating the observations. If you rank by this S prime algorithm, you should find things that are known about lung cancer. You should find the common therapies that you would expect are ranking well, and hopefully elucidating some newer therapies that maybe are overlooked or approaching, we're able to empower that kind of personalized medicine a little bit better. Just briefly, this represents, on the left, it represents running a bunch of those cell lines. Each curve is a unique cell line. One of them we're defining as the reference. The reference would be, again, the non-mutated cell line. So conceptually, we're finding the difference between the mutated and the non-mutated versus the mutated, or you could say the tumor versus the non-tumor tissue. So the purple here at the top is the reference, and we're basically collapsing it into a single value and then finding the difference. And what you see, for example, all these black lines, which each one represents a test, we're seeing a trend where for this drug, whatever the drug was, all of them are having a different curve shifted left, which actually in this case means drug sensitive. All of these would have a delta S score that had a larger magnitude, specifically a negative magnitude because of this minus test at the right. But we're saying that we would be able to find this trend of seeing how all these dose response curves are different using this delta S approach. And that's meaningful because it's saying that the cell lines are responding differently whether they're tumor versus non-tumor tissue. So I want to hand it back over to Paul, who will kind of talk about some of the practical uses for these S indices.
Okay. One of the things that was seen in the last slide was a whole bunch of different curves. And that slide had curves for about 5 different drug cell line combinations. If you're doing screening where you have thousands of curves, it becomes very almost impractical to be able to look at in a single view and sort of understand what's going on. So reducing things to single values becomes very helpful from our perspective. It allows looking at compound target selectivity. You can look at compound inhibition bias, which is working better than the other, cell type specificity. And what we've really focused on is compound ranking and prediction. I know prediction is a very weighted word, but nonetheless, I'll use it anyway in this context. But when we have focused on using delta S as a relative measure and using it in reference to some kind, in regard to some kind of a reference. And that reference can vary. It can be a designated drug. It can be a designated cell line or group of cell lines. It can be a normal versus a cancer, a benign tumor versus a cancer cell, or a cancer in the case that we're pursuing now, with or without a mutation in a tumor suppressor. The output of all of this is information on individual compounds by cell line. We can look at pools of compounds by cell line and compounds by intended target. And all of that with just a single value. So we believe that this, in combination with more traditional values like EC50, will be quite valuable as we move forward and could be generally helpful in terms of looking at new cell interactions and potential therapies or drugs which could be developed for potential therapy. Matthew?
Thank you. Just to reiterate a little bit, one of the things that's an opportunity for us and a challenge for us is that delta S, as highlighted here, can be used in different styles of analysis, right? Saying that you are setting the reference to be tumor versus non-tumor tissue, that's a very beautiful study. Depends on your data set whether you have that. But that's going to be a different use of delta S than, for example, what we talked about before, which was the pooled delta S, where we were looking at populations of cell lines and just looking at the population of cell lines where one gene had one state and that same gene had a mutated state. They're both a delta S algorithm. They both have this kind of reference versus test setup, but they are very different analyses. And so that's some of the work that we're doing as a group. We're looking at how are all the ways that we can apply delta S to do different analyses. And the part of it is that we can accommodate different data sets, but it's active work for our part. Let us take a minute to ask a question that Eric has. Eric has a question which is, how is the data made non-biased? So this is in building the data set. How is the data made non-biased, especially related to cancer stage? I asked Eric, what do you mean by biased? And he responds, if it's stage 4, then there is more mutation. Do stage 4 cells go into the cells in the testing used to build the PRISM data set? So what I'll say is that would probably, as far as data, that would be a tagging question. And there is a fair question, how is the data tagged? How is the data tagged in PRISM? What I will say for PRISM data set, we talked at one point about bringing in mapped data, mapping in the mutations. What we have specifically in PRISM is called a damaging mutations file. And for each cell line in our study, we have the thousands of genes, and there's kind of a 0, 1, or 2 for how many of those alleles are mutated. So we can, for example, look up NF1 in this cell line X. Is it 2 out of 2 damaged mutations? We can that's what we're using as a classifier. Whether we have the designation of cancer stage, I would have to slow down and look at that a little bit closer. I do know that some of the cancer types, for example, in neurofibromatosis type 1, the cancer that we published against was a benign cancer, cutaneous neurofibroma, and then its malignant form is MPNST, malignant peripheral nerve sheath tumor. So there is a kind of a different classification for that tumor when it becomes malignant, and we do have some data that's in that data set where we're looking at this is what the malignant data is and this is what the benign is. But it really it's our algorithm is agnostic. It's really relative to what you're seeing on the screen here. How are you defining reference? How are you defining the test lines? And specific to PRISM, I'd have to look deeper to see if we can make that malignant classification. But what I would be able to tell you is how many mutations it has, how many damaging mutations specifically. We have all of the genes, and we have a count of how many of them are damaged. So we might be able to kind of have a maybe like a pseudo-malignancy metric, but I'd have to see what else they have as far as classifications. All right, so kindly enough, we have reached our end. I want to kind of talk about what we have as next steps and abilities for folks to get involved. So we are currently publishing on this S-prime algorithm. That's coming out in the next few weeks. It's already been drafted, and it's been a team effort for that. Again, focusing on the lung cancer. And we chose the lung cancer because it was a well-known cancer, and we were able to make this kind of scientific statement about tumor suppressors and the synthetic lethality of how you can target things like aurora kinase, those kinds of things. We are looking for feedback, so it's pre-publication. It's not even on anywhere yet. It's all just internal to our group. So if you know people who like biology and they want to read a paper, then we're happy to get some more eyeballs on it. We're now entering that kind of phase. We have a group that meets once a week. This is our computational biology group. It's on this meetup group link on the screen. Meetup.com Petri Dish Bio. They meet every Sunday night, 7:30 p.m. Eastern. And this is where we do the bulk of the work. So this is an all-volunteer effort, and again, we have a series of papers coming up. This is just the first one, or the next one. Future work that we do have coming up is we are trying to apply this S-prime to other cancers, rare diseases. As a community effort, we're able to target rare diseases and have a large impact because major other pharma might not be investing a lot of money, or there might not be a lot of attention. So even a ragtag group such as ourselves, we're able to make a lot of impact scientifically speaking. We also have some really fancy things coming up: network pharmacology, knowledge graphs, machine learning. We're kind of getting into this really fun part of the project where we are able to use some of these analytics tools to generalize our results across cancers. So that's very exciting. We were able to solve hopefully more than lung, but going deeper onto other cancers and build more and more nuanced reasoning for what we're doing. We're also looking for funding, so we do have some publications. Sadly, the publications cost money. They're about $2,500 per paper for a mid-tier paper. So we are looking for that. If you know of any leads for that, please reach out to the organizers. With that, I'm going to take a minute to open it up to questions. Are there questions from anyone online or anybody in the room? Feel free to unmute yourself if you're online.
You mentioned that P53 is a key target, it's suppressed based on many lung cancers. What are the key compounds that can help shore it up? Is that like CDKN1A, or you may have touched on it, but I was just wondering.
Okay, P53 is a very difficult one to work with, and you'll see that when we send out the manuscript, that it tends to be resistant to lots of things. So there are no drugs that I am aware of that directly target P53. So you have to move either upstream of that or downstream of that. So with this picture that's on the screen, for example, moving downstream, you can look at the CDK2, CDK4 inhibitors, because they're in line and downstream from P53. Those also happen to be directly affecting RB1, which is coincidental but could be quite useful from a therapeutic perspective as well.
Oh, very cool. Highlights why this work is important, so thank you.
Absolutely. Other questions or comments? Okay, well, we have time for one last little segue. I'm going to give a shout out to Paul and somebody he knows. We talked a little bit about that CT, computer I'm going to butcher the word pronunciation, but Paul, if you want to give a little call out.
Okay. One of the first known differences between cancers and normal tissue was actually discovered in about 1922. And that was that tumor cells, in general terms, tend to use glucose differently than normal cells. They tend to use glycolysis at the expense of sort of the normal Krebs cycle. They don't process ATP in exactly the same way, and they stick to just processing glucose. So, based on this observation, again going back to the 1920s, a drug was developed that is an analog of glucose, fluorodeoxyglucose, which is taken up into cells, and at high enough concentrations it does kill cells. When you put a radioactive tag on it, in this case F18, which is a positron emitter, cancer cells will selectively take it up, and you can image it. You can take a picture of where the tumor is, both locally and regionally. in metastases. A colleague of mine, Prantika Som, at Brookhaven National Laboratory, was the first one to use F18 fluorodeoxyglucose in cancer. And she was very helpful to me in my career, as was Zvi Oster, who's also listed as a co-author here. He was head of radiology at the State University of New York at Stony Brook. I don't have a picture of him. But nonetheless, I wanted to just have a shout out to them because F18 fluorodeoxyglucose is used worldwide. It is used in imaging for cancer on a routine basis everywhere. And a colleague of mine helped start that. I think that's just very fortunate of me to have an opportunity to work with her and Zvi as well, and I'm quite grateful for everything that they did for me. Again,
That's awesome.
Matthew?
Yeah, just to go back, I think we have a picture of that, right? That's the
It's the colored slide.
colored slide. Yeah, let me go back to that. This one. So that's actually the F18 you were talking about, right?
Yes. So this is superimposed on a CT, but the yellow dots represent the primary tumor. The little circle things are secondary tumors. You might note that there's sort of like a donut effect, and that's frequently observed in tumors because the active rim is more metabolically active than the core. Sometimes in tumors, especially large ones like this, the center is necrotic or hypoxic, so hence a decrease in the intensity.
So Don noticed the connection to the term glucose and asks in chat if there's a connection potentially between diet and cancer, and I think there's something about metabolism at least and cancer that is a connection. Paul?
Don't forget you have a 2-minute reminder on the end of this, so maybe we should just leave it there.
Okay.
There is a connection, and it would probably take too long to go through it this time.
Okay, well, in any case, thank you everybody for coming out. If you have other questions, feel free to reach out on the meetup group. What I'll say is that we will try and post this video. I'm crossing my fingers that the recording went well. Either way, we'll at least share out the slide deck, so look forward to that. Again, these are the ways to get involved, and thank you so much for your curiosity, and have a great evening.
Bye-bye, all.
So do we have a YouTube channel? Where we
Moco Makers has a YouTube channel.
Okay, so we post the recordings on that.